Okay, it's time for part two of the melatonin review. So here we are now going to start going over the melatonin receptors, which are the proteins that are interacting with the melatonin that is produced in your body for it to elicit its effects. So it's not just the chemicals that do effects, your proteins have to be able to interact with those chemicals to actually have that effect or that physiological change take place. So let's begin. So the locations that melatonin receptors are found in the body are as follows: the brain, retina, cardiovascular system, the cardiac ventricular wall, which I'm not exactly sure what that is. So it's the thickest near the, I'll just look at the ventricular wall, and let's look up the ventricle. So, okay, it's one of the two large chambers towards the bottom of the heart. So yeah, these are the ventricles right here, and this is basic physiology stuff, but we forget it, or I forget it explicitly over and over again. So it's nice to go over it when the need arises. So yeah, the cardiac ventricular wall has these melatonin receptors as well. The aorta, which is that giant artery that comes off of the heart, the coronary and the cerebral arteries, the liver, gallbladder, the duodenal enterocytes. So these are the epithelial, I don't want to call them epithelial cells, but they are the basically cells that are near the duodenum in your intestine. So enterocytes, cells of the enteric system. And then you have the colon, the cecum, and the appendix vermiformis, which I have no idea what that is, so let's look that up as well. And I don't see an easy, let's look up the Wikipedia for, well, it looks like they're not going to, okay, the term veriform comes from the Latin meaning worm-shaped. So this is veriform is the worm-shaped part of the appendix. So just keep that in mind. It'd be nice if they just said appendix, but it is what it is. It's also in the skin. It's also in the parotid gland, which, what is that? It's one of the, let's go into the Wikipedia article. It's just one of the chewing glands, or the glands that are in the jaw, and then the exocrine, pancreas, the kidneys, the cells of the immune system. So your immune system also has these melatonin receptors in them. The platelets have these melatonin receptors. The brown and white adipocytes, or your brown and white fat tissues, have these melatonin receptors. Your epithelial cells of the prostate and breast, the ovary granulosa cells, the myometrium, and I think these are muscle. Oh no, they are a part of the female reproductive system. So it's the middle layer of the uterine wall. So yeah, in the uterine wall they have receptors for this melatonin. The placenta and the fetal kidney, and I don't know why, but the fetal kidney reappears over and over again in science. So it might have to do with perhaps fetal tissues are used as model cell cultures just because they're multipotent. But yeah, if people know why fetal tissue or fetal kidney cells explicitly show up over and over again, please leave that in the comments. So in the gastrointestinal system, melatonin receptors are found most commonly in the jejunal and the colonic mucosa. So there are 3 different types of membrane receptors and 1 nuclear receptor. So remember, the membrane receptors, these are receptors that are on the cellular membrane. So these will be on the surfaces of the cell, and the melatonin will interact with the surface of the cell, and then it will transport, or not transport, but transduce that signal from the outside of the cell to the inside of the cell through some mechanisms. And remember from the previous video, that mechanism was using these G proteins. These G proteins are coupling to these G protein receptors on the surface of the cell. And they're called G proteins because they interact with either guanosine diphosphate or guanosine triphosphate. When they're bound to guanosine diphosphate, they are in the off position, and when they are bound to guanosine triphosphate, they are in the on position. So when melatonin binds to these G protein coupled receptors, the receptors will then activate the G proteins on the inside of the cell, and by activating those proteins, they are releasing the GDP and being replaced by GTP, and that turns them on. And when they're turned on, they then go down to elicit their effect in the middle of the cell. So we have 3 of those membrane receptors, and then we have 1 nuclear receptor. So the nuclear receptors will be receptors that actually act upon the nucleus, or the directly dealing with the DNA of the cell. Anyways, the melatonin receptor type 1A, which is also called MT1. This is encoded by human chromosome 4 and consists of 351 amino acids. MT1 receptor constitutes adenylate cyclase inhibition by binding to various G proteins. So this receptor has this adenylase cyclase inhibition. And what is adenylase cyclase? So these are the adenyl cyclases are also abbreviated as AC. These are enzymes with key regulatory roles in essentially every cell. So it's ubiquitous. Every cell pretty much will have these adenyl cyclases, and it is the most polyphyletic, which is I don't know what that means explicitly, but let's look this up. So a polyphyletic group is a set of organisms or other evolving elements that have been grouped together but do not share an immediate common ancestor. So it's the most polyphyletic known enzyme with 6 distinct classes. So I guess that means that there's a lot of variety, or it's the most various type of enzyme. However, let's keep on going. And they all catalyze the same reaction, but representing unrelated gene families with no known sequence or structural homology. Okay, so that makes sense. So they are all doing the same thing. However, they are genetically or related. As they are related to each other, it's very hard to find amino acid sequences or genetic sequences that can link these enzymes together. So they are the most, I would say, diverse class of enzymes that do not have a known amino acid linked to them. Okay, so just keep that in mind. That's what's going on. I don't know how they quantify that. Just know that that is how some people refer to these as. So the best known class of adenyl cyclases is the class 3, or the AC3, and it occurs widely in eukaryotes and has important roles in many human tissues. So all of these adenyl cyclases will catalyze the conversion of adenosine triphosphate, also known as ATP, to 3' 5' cyclic AMP. So this would be, this is what the cyclic adenosine monophosphate is, or CAMP is another word for it. And so you might see this multiple times, but the CAMP is a signal in the cell for various different things. So let me see if, let's not go too far into detail. Just know that by turning ATP, which is one of the energy currencies of the cell, some enzymes, these adenyl cyclases, these will turn ATP into the cyclic AMP, or CAMPs, and that will serve as a signal of some form. Okay, so let's go back to this. So these MT1 receptors constitutes the adenylate cyclase inhibition. Okay, so they will inhibit these adenylate cyclases, and they do it by binding to various G proteins. So remember, the G proteins are the proteins that bind to GTP. So the MT1 receptors are commonly found in human skin. So during the aging process in Alzheimer's disease, the expression of MT1 receptors in suprachiasmatic nucleuses. And what is the suprachiasmatic nucleus? Each of a pair of small nuclei in the hypothalamus of the brain, which are above the optic chiasma. So it's just a little bit, or a little tiny lobe of the brain, it looks like. So let's see if Wikipedia has a... Unfortunately, I don't think they have a good image of where, of where it's located at. Just know it's a tiny region of the brain in the hypothalamus. So it's a part of the hypothalamus. So during the aging process, or people with Alzheimer's, the expression of this MT1 receptor in this small area of the hypothalamus and the cortex, this decreases. So as you get older, or as you get this neurodegenerative disorder, you end up having less of this MT1 that is produced in the brain. So the MT1 receptors reduce the neuronal discharge rate in the suprachiasmatic nucleus and suppress prolactin secretion. So the more MT1 receptors you have, the more you are suppressing prolactin, right? So this is interesting to think about. So some people were talking about how melatonin might be beneficial for people with coronavirus if it's administered. So this would be theoretically one of the possible mechanisms to do that. So we know that prolactin is very, very, very significantly increased in people with severe COVID-19 patients, and so that would be one of the hormones that is going to be dysregulated. And one of the reasons why it's dysregulated would probably be because it has a large role in the immune system functioning properly, or I should say, the more prolactin release, the more of a pro-inflammatory environment you have, right? So if you have these MT1 receptors, they are reducing the neuronal discharge rate in this suprachiasmatic nucleus, and this also will help them. I don't know if these two things are linked, but the release of prolactin will be suppressed by these receptors. And so by proxy, you could also argue that by activating these receptors with melatonin, that you would also reduce the amount of prolactin that is being released. So what I'm going to do is, yeah, I like that. So I'm going to have that ready for later. So that article is right here. So just so you know, these MT1 receptors that are on certain, like they're on your skin, they're in your brain, they are activated by melatonin, and one of the effects that they can do is suppress the release of prolactin. So that's good to know. So the melatonin receptor type 1b, also known as MT2, this is encoded by human chromosome 11 and consists of 363 amino acids. MT2 receptors creates Adenylate cyclase inhibition by binding to various G proteins, so it's a very similar process that is going on between these two. And additionally, it inhibits the soluble guanyl cyclase pathway. So it doesn't just inhibit the adenyl cyclases, it also inhibits the guanyl cyclase pathway. So what is the guanyl cyclase pathway? So here are the guanyl cyclases, or this is an example of the catalytic domain of human soluble guanyl cyclase 1, and it will undergo, it'll take these GTPs, remember this is the same molecule that associates with our G proteins, it'll take these GTPs, guanosine triphosphate, because there are 3 phosphates, it'll take this, and then it will cyclize one of the phosphates, and now this is cyclic GMP, or cyclic guanosine monophosphate. So, and the reason why it's called 3' to 5' is that it comes from the 3' carbon and it goes to the 5' carbon. So just so you know, that's the reason why it's called that. So that is the guanylate cyclases. So these MT2 receptors will inhibit the soluble guanyl cyclase pathway as well, not just the adenyl cyclase pathway. And through melatonin receptor activation, the adenylate cyclase inhibition occurs, and the production of cyclic AMP is reduced. So the activation of both MT1 and MT2, both of those will help inhibit cyclic AMP production. So in the skin, the MT2 receptors are located within normal and malign melanocytes. So these are the cells that are producing melatonin. So let us look about what a melanocyte is. So the melanocytes, these are these darker cells that are within the cell that are producing melanin. So they are the melanin-producing neural crest-derived cells located in the bottom layer, which is the stratum basale of the skin's epidermis. So they're in the bottom layer of the epidermis. Okay. So just, you know, that's where they live and exist at. So these MT2 receptors, or these melatonin receptors 2s, they are located within the normal and the malign melanocytes and the eccrine sweat glands. And the eccrine sweat glands, just know that these are a type of sweat gland, and I believe they are the sweat glands that they might be the ones that do release a lot of pheromones. Let me just look at this real quick. So these are the eccrine sweat glands, and they are the major sweat glands of the human body, and they are found virtually in all skin. And then but much less on the trunk and the extremities. In other mammals, they are relatively sparse, being found mainly on hairless animals. And yeah, so I know that they have the eccrine glands, and there is another type of gland, but I can't find it right now. Just know that there are multiple types of sweat glands. These eccrine sweat glands are the most common ones on the body, but there are different ones, and I can't remember what they are exactly. So MT2 receptors inhibit GABA A receptor related functions in the hippocampus in rats. So what is GABA? So this is gamma aminobutyric acid, which is a related, or actually, I'll just read through it. So it's a chief inhibitory neurotransmitter in the developmentally mature mammalian central nervous system, and its principal role is reducing neuronal excitability. So this is going to be a molecule that if it's in higher concentrations in the nerves, it's going, or in the nervous system, it's going to prevent the neurons from firing as easily if that is a neuron that has the receptors for this GABA. So its principal role is reducing neuronal excitability through the nervous system, and GABA is sold as a dietary supplement at times. So as a neurotransmitter, it acts as an inhibitory, it inhibits at the synapses in the brain by binding to the specific transmembrane receptors. So it's not just going to suppress all of your neurons. That's not exactly how it's going to work. It's going to bind to certain receptors that is only on a subset of the neurons in your brain, and by binding to those receptors, they are going to suppress the excitability of the nerves that have those receptors. So it's not going, if you just take GABA, it's not going to just completely dull your entire brain. It's only going to attack, for lack of a better word, it's only going to attack the GABA positive, the ones with the GABA receptors. So just so you know, there's GABA A, which is this, and then there's also GABA B receptors. So just know you have multiple receptors for the same chemical. Regardless, melatonin 2 or MT2 receptors will inhibit GABA A receptor related functions in the hippocampus. So if you could imagine, if you have these MT2 receptors and you are given melatonin, it will actually inhibit these GABA responses. So remember, GABA reduces excitability, right? But these melatonin receptors will inhibit that GABA, those GABA cellular signals, right? And so if you activate MT2, you end up increasing the excitability of those hippocampal cells, right? So if you could imagine, what would this equivalent, or what would be the downstream effect of this? The downstream effect would be, if you are producing melatonin, then that melatonin will make your hippocampus more active. So when you are sleeping, or when you haven't seen sunlight in a while and you're starting to get drowsy, you're starting to get sleepy, your hippocampus is now becoming hyperexcitable. It's now more excitable. And so that might be some of the reasons why if you are drowsy or have lack of sleep, you might start seeing things that aren't actually there, So the hippocampus, remember, that is associated with the conversion from the short-term memory to the long-term memory. So that's just one of the many functions that the hippocampus does. So just know that if it's more active, just know that it is more active when you sleep, or when you are in the process of trying to go to sleep. So in Alzheimer's disease, these MT2 receptor expression is reduced as well. So remember, in MT1, in Alzheimer's disease, will reduce these melatonin receptor 1s, and in Alzheimer's disease, it will also reduce these MT2s. And these MT2s receptors are involved in antidepressant activity. So remember, as we were saying earlier, MT2 will suppress these GABA proteins, right? Or I shouldn't say GABA proteins, but these GABA receptors. It'll inhibit their activity, and what those GABA receptors do is they will make it makes it harder for your nerves to fire, right? It's just more difficult for those nerves to fire, and therefore, let's say you are someone who your nerves just really do not want to fire, or they are difficult to fire. It's hard to get you excited. That could be a form of depression. That could be a form of a suppressed neuronal response, right? So these MT2 receptors are involved in antidepressant activity, right? So if these aren't working, you might not even, you might have depression as a result from them. So these MT2 receptors contribute to the pathophysiology and pharmacology of sleep disorders, anxiety, depression, Alzheimer's disease, and pain. So MT2 receptors may be the new target for development of hypnotic agents. And I believe these would just be chemicals that help you undergo or do hypnosis. They're commonly known as sleeping pills and are a class of psychoactive drugs whose primary function is to induce sleep and to be used in the treatment of, or induce sleep and be used in the treatment of insomnia. So hypnotic, I guess, try to just get people to sleep, right? So MT2 receptors are responsible for the anxiolytic or the anxiety-reducing effects of melatonin. So as you are giving, if you give melatonin to someone, it ends up reducing their anxiety, and the primary mechanism of that is through the MT2 receptors. So apparently the MT1 receptors do not play as much of a role in that, which makes sense if the MT2 receptors are the ones that are dealing with the excitability of the hippocampus, or they're dealing with the suppression of these GABAergic neurons. It makes sense that those are the ones that are going to be more associated with trying to get your brain to turn on and off during night. So it makes sense that these MT2 receptors are the ones that are associated with that effect. Whereas MT1, you know, that's in the cells, or in the skin cells, right? So that's a big part of them. And then what else was the MT1 associated with? Yeah, the suprachiasmatic nucleus. So I guess there are some brain parts that also express MT1 as well. But yeah, it seems as though the MT2 is the reason why we take melatonin in the first place in regards to it being a sleeping agent. So those are the first two. Oh, I should say, and then the MT2 receptor ligands have more powerful hypnotic properties when compared to the non-selective MT1, MT2 ligands. So yeah, this is what I was saying earlier. If you have a drug or a pharmaceutical or a chemical compound, even if it's coming from nature, that is administered to someone, the ones that target MT2 are going to be the ones that elicit more of this hypnotic or sleep-inducing effect. So, and the third receptor, or another receptor, is the ME11C, or MTNR1C, and this is not present in humans. So I don't know why they're talking about it here, but just know that in fish, amphibians, and birds, they have this additional membrane-associated transmembrane receptor for melatonin, and in chickens, the rhythm of this receptor is the opposite of MT1 and MT2, and its level is highest at daytime and lowest at nighttime. So what this might actually be doing as well, 1 second, it's closely related, right? It's probably a related protein. However, related proteins are sometimes just change a few amino acid sequences or change the protein enough, and you end up with a protein that has the exact opposite function, right? And so apparently in the fish, amphibians, and birds, or I should say in chickens, this rhythm is the opposite of MT1 and MT2. Okay, so just know that that is what might be happening. It would still technically be an oscillator, but it is eliciting its effects on the oscillator at probably a different part, or a different part of the wave function, if that makes sense. So moving on to MT3, this is the quinone reductase 2 enzyme. So what is quinone? That is, well, I should say there are a lot of different quinones, right? And so quinone just means you have an aromatic ring, and so here it's a it has to be a 6-membered ring. So this would be benzoquinone. So it is normally a benzene, or it was originally a benzene, which is when you have 6 carbons in a ring that have alternating double bonds, which ends up equivalating to 1.5 bonds between each carbon because it's symmetrical and aromatic. Regardless, benzoquinone maintains its aromaticity. However, you have 2 ketone groups, and that's what a quinone is. So they are a class of organic compounds that are formally derived from aromatic compounds such as benzene or naphthalene, and this is naphthalene if you want to look at that. And it's done through the conversion of an even number of these how do I phrase that? I'll just say these aromatic carbons. An even number of aromatic carbons go from their normal aromatic state, where you have 1.5 bonds to another carbon. It goes from that into being a ketone group, or a carbon that is doubly bonded to oxygen. And there is necessary rearrangement of the double bonds, resulting in a fully conjugated cyclic dione. So a dione would be a chemical that has 2 ketones in it, and then fully conjugated would mean all of the carbons have a particular hybridization to them. Don't worry too much about it. Just know that it maintains some of the similar structures of the original benzene. So, or the original aromatic compound. So the class includes some heterocyclic type compounds. So just so you know, these are what the quinones are, and there's actually a large range of what quinones exist in nature. So these quinone reductase 2 enzymes will take a type of quinone, and then they will reduce it. I don't know what particular reducing agent they use. It would probably be FADH2 or NADPH. Regardless, they will add electrons, or they will reduce the molecule to become more electron rich, if that makes sense. So let's look up the Wikipedia. Okay, so it does use NADPH or NADH, depending on what chemical is nearby. So this is what this NADPH dehydrogenase quinone 2 enzyme looks like. And so, it's a detoxification enzyme which can carry out 2 or 4 electron reductions, and its mechanism of reduction is through covalently bound zinc ions, and it is often co-crystallized with FAD, which is another, that's an oxidizing agent. So it's not a reducing agent, it's an oxidizing agent. Anyways, so yeah, this is what we are talking about when we are talking about these quinone 2 reductase enzymes. So that is what MT3 is. MT3 will do this chemical process, but that is independent of its melatonin binding. So this enzyme belongs to the reductase group, which are involved in prevention from oxidative stress. So they are antioxidant. How do I phrase that? They are antioxidizing enzymes. So they'll take oxidants in the environment, and they will basically sequester them so that they don't do damage to the other biomolecules of the cell. And they prevent oxidative stress by inhibiting the electron transfer reactions of quinones. So quinones can be active in redox reactions. So if you reduce them and then make it so that they can no longer participate in these redox reactions, that would be a way to sequester them and basically improve the health of the body or the cell that they are a part of. Anyways, this enzyme, also known as the Mt3 receptor, is located in the liver, kidney, heart, lung, intestines, muscles, and brown fat tissue. So, and just, you know, the brown fat tissue, this is the tissue that has high levels of mitochondria, and they basically, mitochondria, they're the powerhouses of the cell, right? But mitochondria, their normal job is to produce chemicals, produce ATP, or produce the chemicals that can function as energy currency of the cell. Now with and it accomplishes this by having a voltage across the membrane of the mitochondria membrane. So the mitochondria membrane has a very strong, or a large voltage that goes between the inside of the mitochondria and the outside of the mitochondria. And this voltage is what is used to basically move the machinery that is necessary to create these high energy compounds. And by making these high energy compounds, they can serve as currency for the cell to use for different biological processes. Now the problem, or I shouldn't say the problem, but what brown fat tissue does is it says, screw it, I'm not going to make energy currency, I'm just going to make heat. So rather than creating a product that can be used as energy currency within the cell, it just wastes that energy current, it wastes that voltage, and by wasting that voltage, you are releasing heat into the environment. And so this is used by babies, for instance, in order to keep warm. While they still don't have a lot of muscle mass and a lot of mass in general, their brown adipose tissue can just burn through the fat or burn through the lipids in order to produce heat. Just so you know, the brown fat tissue has these melatonin 3 receptors on them. Anyways, it is a detoxification enzyme, and there's evidence for its involvement in regulation of intraocular pressure. So this would be the pressure of your eyes, I believe. But let's look. So it's the fluid pressure inside the eye. So yeah, just with the fluid pressure inside the eye may or may not be influenced by these Mt3 receptors. So let me drink. It's just lemonade. So that is that. And then 5, we have the RZR ROR alpha, which is the retinoid-related orphan nuclear hormone receptor. So what are let's just look up what this is real quick. So the RAR-related orphan receptor, this is a member of the nuclear receptor family. So the nuclear receptors, these are receptors that after they bind their ligand or bind what is important to them, they will either interact or stop interacting with DNA. And their interaction with DNA will determine whether or not they are expressing genes or inhibiting genes. It basically just changes genetic expression in that particular cell. So there are 3 forms of the RORs, the alpha, beta, and gamma, and each is encoded by a separate gene. So they're specifying separate gene because sometimes you can have the same gene be spliced differently in the mRNA version of the gene, and that differential splicing can pop up as different proteins. However, we have 3 different ones, and they are from distinct genes for this particular receptor. Anyways, the RORs are somewhat unusual in that they appear to bind as monomers to the hormone receptor or hormone response elements, as opposed to the majority of other nuclear receptors which bind as dimers. So this is interesting to note. So most receptors, or a lot of receptors in the cell, are proteins, but these proteins usually have to dimerize in some way in order to interact with DNA. And a lot of that sometimes has to do with its ability to clamp onto each side of that double helix. So each side can have part of the protein, and a lot of times to get that proper shape, it requires dimerization of some form. However, these RAR-related orphan receptors do not need that. They can bind just as monomers, which is pretty unique. So, and they bind the DNA elements, which are called ROR response elements. And remember, element very frequently will refer to a genetic sequence either on DNA or RNA that a protein can bind to. And when it's a response element, that means that protein is going to bind in relation to a response, either coming off of it or coming onto it due to a response. So that's why they're called response elements. So while the identity of the natural ligands for RORs remain controversial, similar to the liver X receptor would just be another example of these nuclear receptors, it appears that the RORs are activated by oxysterols, so these would be steroids, or they would be related or the derivatives of cholesterol. So that might be what activates these orphan receptors, but we still don't know exactly what is activating these orphan receptors. So that is why they are called orphans, right? They're orphans in that they don't know what their parent ligand is. So let's see if there's anything else that we should, just know that there is some differential tissue distribution. But yeah, these are the ROR or RAR-related orphan receptors, also known as RORs, which we love our acronyms in biochemistry, right? Anyways, with this receptor, melatonin binds to the transcription factors in the nucleus, which belong to the retinoic acid receptor superfamily. So that's why they're called RARs, or retinoid related, right? So they're related to these retinoic acid receptors. So if you think about vitamin A, that would be retinoic acid. So let me just double check that. Yeah, so it's a morphogen derived from retinol. So if you take retinol, which is vitamin A, it gets converted into retinoic acid, which is what is binding to a lot of these proteins as one of the cofactors. So the following are described for retinoic acid receptor superfamily variants of ROR alpha. Yeah, don't worry too much about all this. These are just the different ones. So I wish there was more about this. Just know that melatonin is able to bind to the transcription factors in the nucleus that do bind to this ROR alpha. Anyways, continuing, I wish, yeah, that might be something that people want to look at. So how melatonin will influence the genetic expression through means of this ROR alpha receptor, that might be something important or useful for a lab to look into. So there's also the melatonin-related orphan receptor, X-linked orphan G protein coupled. Mouthful right there, right? So it is an X-linked inherited receptor which binds to G protein. So remember, this is G protein coupled. So remember, the G proteins, these are proteins that will bind to GTP or GDP, right? So this is a different protein that binds to melatonin that is also able to interact or be coupled with G protein, right? And they call it X-linked because it's on the X chromosome. So remember, the X and Y chromosome determines your sexual dimorphism, right? And so your sexual dimorphism will be determined whether or not you have 2 or 1 X chromosomes with or without a Y. And this, this is the standard distribution. However, there will always be exceptions to the rule, right? Anyways, this is X-linked. It's an orphan G protein, so we do not know what its real ligand is supposed to be. But it is, just so you know, X-linked, it's orphan, and it's a G protein coupled protein. So, and it is an ortholog of MEL-C or MEL1C, which is just a closely related protein, and this one is found in non-mammalian living creatures. So its gene is located on the X chromosome, and it consists of 618 amino acid residues, so it's a larger one, and it is present in all mammals, including humans, and it does not have the characteristics of binding to melatonin. However, it is effective in binding of melatonin to MT1. GPR50 is not present in birds and fish and is located in the brain and periphery, and its natural ligand has not been defined yet. This is why it's still called an orphan. So it was reported that a deletion mutant in GPR50 might have been associated with bipolar disorder and major depression. So perhaps a mutation in this will cause the inability for melatonin to bind to MT1 correctly, possibly. So GPR50 has no affinity for melatonin. However, when it dimerizes with MT1, it inhibits the melatonin signal. Okay, so yeah, this makes sense. So this is inhibiting the signal of MT1. So it is one of the suppressive or counteracting or negative feedback loops that might exist in the cell. So GPR50 has other functions apart from melatonin. GPR50 interacts with the neurite outgrow inhibitor, or NOGO-A, and TIP60, which is a glucocorticoid or one of the steroid receptor signal coactivators, and histone acetyltransferases. So histone acetyltransferases are just proteins that can add acetyl groups to histones, which are the spools that help organize the DNA within the cell. Don't worry too much about that. So, yeah, so that is what GPR50 may play a role in. And let's go to the Wikipedia article to see if there is anything more that needs to be added. So just know that certain mutations of GPR50 in females are associated with an increased risk of developing bipolar affective disorder, major depressive disorder, and schizophrenia. and other GPR50 gene mutations are associated with higher fasting circulation triglycerides. Oh, I think I've gone over this one before. So yeah, in the previous video I went over that. So after MT1 and MT2, which remember those are the surface receptors, after they adhere to the cell surface, they create their effects through these G proteins. And activation of the MT1 receptor leads to inhibition of CAMP formation, which is stimulated by Forklin. And what is Forklin? And let's get Wikipedia up because Wikipedia's... Is it Forskolin? Yeah, perhaps that is a typo where there should be an S in there. But this is Forskolin, and it's commonly used as a tool in biochemistry to raise levels of cyclic AMP, also known as CAMP, right? And remember, this is what cyclic AMP looks like. So activation of the MT1 receptor leads to inhibition of CAMP formation, which is stimulated by Forklin. So in this particular model, we add Forklin, hoping to cause greater expression of CAMP, but if the MT1 receptor is also activated, it will suppress that effect. So together with inhibition of the protein kinase A, also known as pKa. So pKa is, I believe, one of the kinases that is activated by CAMP. So let's look at this. So protein kinase A in the family of enzymes whose activity is dependent on cellular levels of cyclic AMP. So yes, so by having cyclic AMP present in the cell, this will activate this protein kinase A. And by activating this protein kinase A, it's then going to add phosphate groups to different proteins to turn them on or turn them off, right? So what is its role? Let's save it. So the pKa phosphorylates proteins that have the motif arginine X serine, which are exposed in turn, which deactivates the protein. So it looks for this arginine blank serine code or amino acid sequence on proteins, and when it finds this sequence, it will add a phosphate to it, and by adding a phosphate, it'll deactivate that protein. So just so you know, that's the general mechanism of how these pKa's work. So when you inhibit the CAMP formation, you are also inhibiting the protein kinase A activity. And this is done through the MT1 receptor activation with melatonin. So similarly, activation of the MT2 receptors leads to inhibition of CMP formation, which was also stimulated by Forklin. So they did the same thing with the MT2 receptor, and it was suppressed. But on top of that, additionally, it inhibits the formation of cyclic GMP. So not only cyclic AMP, but cyclic GMP will also be reduced. So while membrane receptors are basically located in the central nervous system, these RZR-ROR alpha is located at both the periphery and the brain. So just so you know, the peripheral nervous system is separate from the central nervous system, and a lot of times there'll be differential expression of the receptors on the neurons. And so this RZR, so these MT1s and these MT2s, they seem to be more highly expressed in the central nervous system versus the peripheral nervous system. But these RZR-ROR, which remember is a nuclear receptor that will interact directly with DNA, this one seems to be located at both the periphery and the brain. So interesting. So membrane receptors and their specific agonists. Agonists are ligands that are able to bind and activate a receptor, in contrast to an antagonist, which those are ligands that can bind and prevent the actual activating ligands from binding. These membrane receptors and their specific agonists are associated with the circadian rhythm. So these receptors somehow are able to tune the oscillation of the cells within your body, and by tuning that, your body is able to live within or live in harmony with itself, or I should say live with the same time zone. So you don't have different time zones in the body, so you need everyone to be on the same clock. So the membrane receptors seem to be associated with the circadian rhythm, but in contrast, this RZR-ROR, which is a nuclear receptor, this seems to be responsible for the immunomodulation at the periphery, cellular growth, and differentiation of bone. So activation of the protein kinase C alpha is a critical step in the formation of melatonin effects. So Yeah, just know that this is another cellular signaling molecule. So, development of pharmacological agents which are effective on the Mt3 receptors may be associated with antihypertensive, which hypertension, this would be high blood pressure. So antihypertensive medications would lower your blood pressure. So these development of these drugs against, which are effective on these Mt receptors, could be associated with the antihypertensive, the anti-cancer, and the immunostimulant effect. Or they may facilitate falling asleep, right? So it could just be the simple falling asleep thing that you want to go for. So in addition to its anti-inflammatory effects, its immunostimulant effect is an undesired situation in autoimmune disorders, and metallonergic drugs may be contraindicated in such patients. And contraindication means suggests or indicates a particular technique or drug should not be used in the case in question. So if there's a contraindication, that means do not apply this drug to this particular set of conditions. So if the condition is someone has an autoimmune disorder, perhaps the administration of melatonin might not necessarily be good. However, it could be good. Who knows, right? So there are a lot of, if something falls out of whack, what drug works for one condition might actually have the opposite effect for a different condition. So just so you know, it's all this, biology is a noisy field, right? So getting this all straight is pretty important, which is why we have FDA trials, right? Anyways, for example, melatonin aggravates the symptoms of rheumatoid arthritis by stimulating pro-inflammatory cytokines. So that is interesting that you, so when a lot of people talk about melatonin, we talk about usually how it has this nice calming, relaxing, suppressing effect, and how it can help with the immune system. But it seems that in special cases, especially for, say, people with rheumatoid arthritis, melatonin is actually screwing them over if you give them more melatonin. So melatonin is not just anti-inflammatory, it can be pro-inflammatory in certain conditions. So keep that in mind. So with this, I might do the drugs later, but I think with that we've gained a comprehensive, or at least a more comprehensive understanding of how melatonin works. And so with following videos that I do on this particular chemical, you would be able to go back to these videos and basically understand, okay, how is the melatonin fitting into these receptors, and then by fitting into these receptors, they are causing or eliciting an effect on other proteins that are in the cell or outside of the cell, right? So with that, thanks for watching, and if you have any questions, leave them down in the comments, and I will leave the citation for this in the description if you want to read this paper yourself. So with that, thanks for watching, and hopefully I will see you tomorrow.